Hi guys, David, Almost Daily Science. So today I want to look at what may turn out to be the most disgusting science research report that I will have ever read in my life. And the reason why I'm reading it is probably morbid curiosity. I did read the headline of it and I thought, wow, that's appalling. I need to know more. So I do want to warn you that this might be more disgusting than you're ready for, so just be aware. So the research report is about a fungus that uses chemicals to trick male flies into mating with infected dead females. The article says that prior research has shown that some types of fungus can give insect victims what has become known as summit disease, in which a victim's nervous system is infected and the unwilling creature begins climbing to the highest vantage point possible. Once there, the wings are spread wide and the victim begins spewing spores. So that's pretty disgusting. But this research does not stop there. It takes it one step further by having its female victims also emit chemicals that sexually attract mates. In studying the fungus Entomothora muscae, the researchers found that it was capable of infecting other insects, primarily houseflies, with summit disease. Airborne spores land on female victims and penetrate her skin. Soon they invade her entire body, including her nervous system and brain. Chemicals produced by the spores incite the female to begin climbing until she reaches the highest possible point, such as a leaf on a tree. Then she opens her wings and dies. Meanwhile, the fungus covers her body with little spore-filled cannons. At some point, a male happens by, and when he touches her body, the cannons fire, filling the air with spores, ready to infect others in the vicinity. They should maybe make like a horror movie based on this. Maybe they should not make a horror movie based on this, actually. So let's look at the actual research paper itself so that we can dig a little bit deeper into the experiments and results. I should say that this is not certified by peer review. This is what is called a preprint, so it hasn't gone through peer review yet. But the paper is called "A Pathogenic Fungus Uses Volatiles to Entice Male Flies into Fatal Matings with Infected Female Cadavers." The abstract says, to ensure dispersal, many parasites and pathogens behaviorally manipulate infected hosts. Other pathogens and certain insect-pollinated flowers use sexual mimicry and release deceptive mating signals. However, it's unusual for a pathogen to combine both this host manipulation and sexual mimicry. The paper says the evolution of specific mate recognition systems like pheromones and things like that is often central for successful sexual reproduction. Once males and females have located each other, individual mating preferences or competition for access to mates may lead to suboptimal decisions during courtship and mating. I don't know if it gets more suboptimal than being tricked into mating with a cadaver that then uses you as a vector for spreading a horrifying fungus. But they're talking more generally here. So the willingness to mate is, for example, exploited by certain insect-pollinated flowers, which use sexual mimicry to attract pollinators by resembling the opposite sex visually and/or chemically. So we know about that. That's a key function in certain pollination processes. So this fungus represents a unique example in that it not only behaviorally manipulates its immediate housefly, but also appears to manipulate uninfected conspecifics. Prior to death, infected houseflies are manipulated to seek out elevated positions and die with wings spread out in a specific posture conducive for active discharge of infectious fungal conidia, which occurs primarily within the first 24 hours post-mortem. So that's kind of what we were talking about before. The article was talking about summit disease, which I guess is fairly well known and understand. And it says that male and female flies in the vicinity may be infected from being directly hit by the spore, or via contact with already discharged spores that form a halo on the surface surrounding the conidia shooting cadaver. So that's pretty gross. Remarkably, the behavioral manipulation continues after death of female hosts, as the fungus appears to use sexual mimicry to lure healthy males to attempt mating with these cadavers, and that's going to spread the spores even more. So this is pretty interesting just about how houseflies mate, and since, as I said before, we're just diving into the grossness, we'll just go ahead and talk about it. In houseflies, courtship starts with the male jumping on top of the female in a so-called mating strike and placing his legs at the base of the wings of the female, which in turn instantly spread her wings horizontally from the body, resembling flight position. Both males and females are considered as choosing sex as the choosing sex in houseflies because males vary considerably in their mating efforts, So I guess they'll try harder in some instances than others if they're perceiving the female to be more desirable. And I don't know if that's the right word for houseflies. And females can exert mating choice by kicking off courting males. Although housefly males are able to distinguish between male and female cadavers covered in infectious conidia, the infectious spores, males do not discriminate the fungus-swollen female cadavers and appear to readily initiate courtship and mating strikes. Okay, so in terms of the results, they're saying that we first performed mating activity experiments to verify increased male mating attempts towards the E. muscae killed female cadavers. The first observation is that males neither spent more time in the vicinity of nor physically touched fungus-killed cadavers more often than control cadavers. So I guess they just had some dead female flies, and then they had some infected dead female flies. And there was no, at least initially, there was no difference. That like the males didn't really seem to be attracted to either of the corpses. So good on the males for that. This was irrespective of whether the cadavers were in the early 3 to 8 hours post-mortem or late 26 to 28 hours post-mortem sporulation stages. However, male mating attempts increased as the cadavers proceeded to the late stage of sporulation and were not surrounded by a halo of infected conidia. This increase in sexual attraction appeared sex-specific. Males mated more frequently only with female cadavers, confirming previous observations. Interestingly, when we allowed a male to choose between a control and infected female cadaver present together in a small arena, we did not see a significant difference between infected and uninfected cadavers in the number of mating attempts. Okay, well, that's interesting. Maybe that just implies that flies are just kind of are gross, which I think we knew already. However, a significantly higher number of total mating attempts to either of the two cadavers in the arena was observed if just one of the two cadavers was in the late stage of sporulation. So that's interesting. So I guess just the presence of the spores, like they're breathing in the spores, it just kind of like put the male flies into kind of like a mating frenzy. And then once they were in that frenzy, they didn't really distinguish between a female that was infected versus a female that was uninfected. Flies are still gross though, because remember both of the flies are dead in this instance. So looking at figure 1, we see this first figure this is a healthy male fly attempting to mate with a female cadaver, and fungal growth is seen as white bands extruding from the abdomen of the dead female. So I guess this stuff here, that's the fungus growth, and I think that these white spots all over the place are probably the fungal spores, and you can see the fungal spores are kind of like all over the place. I hope it was worth it. And then in terms of the actual data, we have in subfigure B, this is male mating attempts towards uninfected freeze-killed or infected E. muscae killed cadavers in the early 3 to 8 hours post-death and late 26 to 28 hours post-death sporulation stages. So that's these first two. You can see the blue dots. This is the mating attempts or the counts on the y-axis. So I guess in this research, they probably had cameras on, and they were probably then deconstructing the video afterwards and just probably counting the number of times that the male flies would try to mount on the dead females. So it looks like the blue are these EM, and then the infected are the orange, the plus EM. And it looks like in the early sporulation stages, there wasn't really any difference in the number of mating attempts between infected and uninfected corpses. However, it looks like as they get to the later stages, it says late sporulation cadavers, you can see that the plus EM group is showing significantly more mating counts. We're up to like 30 mating counts in some of the trials, versus the best trial for the non-infected had less than 10. And just in general, we're seeing a lot of 10s in terms of mating counts instead of maybe like 1s and 2s, I think is what we're seeing here. So it does look like in the late sporulation stage, you start to see this difference in mating preference, and just more mating in general is happening. And then in this other experiment we were talking about, shown in C, basically in this one, a male is put in the presence of both infected and uninfected cadavers, and just overall, just being in the presence of the sporulating and the chemical releasing females just made them mate more in general. Poor sods. So they went on to analyze some of the volatile chemicals that were being emitted by the different corpses. And you can see, for instance, this column, so we have these tentatively identified organic compounds that were collected from the head space over the where the flies were. So they kind of like collected the air that was in the presence of the different types of dead flies, and then they analyzed that using gas chromatography. And you can see that we have the negative EM female and the negative EM male. And just generally, you can see that there's a lot more minuses here, which means that different chemicals listed here were not present, whereas the infected male corpses and female corpses, you see a lot of pluses here. That means that a lot of these compounds that were not present from just the normal uninfected corpses were being emitted by these specially infected corpses. They then also took samples. Somehow, I didn't read the details on this, and so I'll have to rely on peer review eventually when it comes out to see if this was legitimate or not, because this does seem a little dicey. They took samples of the antennae of the male houseflies, and they tested the presence, basically, of some of these compounds using, again, GC. So I think they probably took the antenna themselves and then ran those on this analytical technique known as gas chromatography, and then they showed that there were trace amounts of these chemicals actually present in the antenna, which indicate that the antenna of the males were actually picking up these special scents. And so the hypothesis is, or to bring the full idea around, the idea is that these chemicals are being released by these infected cadavers, and the houseflies, the male houseflies, are actually receptive to that. They're picking up on these chemicals, and presumably that's what's altering their behavior and making them mate more when they're in the presence of an infected cadaver instead of an uninfected cadaver. So anyway, I would be curious to know if this is in fact the most disgusting research paper that you've ever seen. If not, go ahead and tell me what the most disgusting one is that you have seen, and we'll go from there. Anyway, I hope you did get a laugh out of it. I did think it was funny, even if it was gross. So if you enjoyed the video, please go ahead and like and subscribe, and I hope you'll join me again in the future soon. Bye.